Hi, how are you? My name is Namjoon Cho. I'm the professor at School of Material Science and Engineering at Nanyang Technological University. Thank you very much for this talk invitation. The title of my talk is investigating of a biorecognition processing using force spectroscopy AFM with molecular functionalized tip. So as you know, the POC system developed very nice non-contact AFM system, scope microscope and spectroscope. And we are try to using different application other than right now it's very hot technique for the semiconductor machine evaluation for the morphology and so on, as a background of material science and chemical engineering, as well as medical school, which is gastroenterology. One of my big study is hepatitis C virus, which we got this subject got the Nobel Prize this year, thanks to the Charles Rice and then collaboration with Jeffrey Glenn, which is my former collaborator at Stanford University. So, our group is basically, I always think, there is two different type of research, which is applied and not yet applied. So, we try to using this kind of technology to apply science, which is in this particular case, I'm going to give you the example of other than semiconductor industry. What can you do with the atomic force microscope technique, which we are using it for drug screening platform? So this is my group, and then it's a brilliant people, the very nice people, especially if you have the people already graduate and get the PhD, postdoctoral fellow, go to different place as a faculty member, and also go to industry and start a company and so on. So this is the funding acknowledgement I start with. So if you actually see the funding in here, you can clearly see that a lot of my research relate with the infectious disease right now, especially for the COVID-19 pandemic, and that's what I'm working on as well. And also targeting assembly and function of the Hep C, which is replication, and influenza infection transmission, and so on. So it's a very exciting topic for me, and then definitely I'm hoping that one day I can contribute something. So this is the basic background. I did the material science, chemical engineering, as well as a subtopic of infectious disease. At the time of the year, it was very rare, study infectious disease from engineering viewpoint. Then I got the award for the National Research Foundation, and 2011 moved to the NTU as a Nanyang Associate Professor. And then also I served as a consulting associate professor at Stanford University, principal investigator at Singapore MIT Alliance for research and technology, and so on and so forth. So, now currently I am the Nanyang professor, and then also the MRS professor. So some of the my studies, we basically share the vision of how do you people using engineering tool to approach new area of science. That's what we are often doing it. And then many of the innovation and global impact is published in the various of media outlets that you can actually search it for. And then one of the technique we use is AFM. So what we're doing is conventional biorecognition process is a biologic assay people using it. Like, for example, you have a transducer, which is a biologic component you are putting in, and also you want to detect sample like right now current diagnostic, right? You do the diagnostic COVID-19 and so on. It's a PCR based or rapid diagnostic based and so on and so forth. But that biosensing process or biorecognition process is you need to have a transducer and then you need to get a signal. Can we do exact same thing using very, very detailed process like or precise or very, very high resolution process, which we are taking using AFM because it's one of the technique we using previously is, typically confocal microscope, SPR, QCMD, Western blot, in the just conventional analytical tool, as well as a Western blot tool. But however, not many people using the atomic force microscope, because I believe the origin of the atomic force microscope is from the viewpoint of physics, not from the biologic side. And then we try to tackle this, wide open applications part. And then, as you know, the basic principle of AFM operation, which I'm not going to go in detail. However, you also have manipulation of the teeth and approach contact and pull out adhesion. In those processes, you can using it as a force spectroscopy. So basically, if you're using those AFM teeth with the precision control, like non-contact mode developed by or originate by the PAC system, you're able to actually modify the lab as you're showing in here, and then schematic graph, and then you can basically modify your AFM teeth, single or multiple teeth. Then you can put the linker, then you put the sample objective, try to see the interaction between either membrane or transmembrane protein within the membrane and so on, so that you can actually capture very precise force between the sample and the subject itself. That's what we're using for the AFM, because it's very much precise. The first result is basically how are you going to correlate single molecular interaction compared with the assembled average measurement of the peptide absorption on different organic material. That was the very interesting study, and then we already published in the 2016. And using that, what we do is we compare this data with the QCMD technique data, so that we correlate with our results, so that we combining the experiment technique allow us to correlate changes in the peptide binding affinity. And using AFM, one of the advantages, you can actually measure the single detection. However, using QCM, quartz crystal microbalance, you are basically measure the assembled signal from the whole surface. That's the main different thing. So by doing that, compare with the QCMD data, you can actually see how the peptide is interact with depending on the surface. And then here showing the silicon oxide, titanium oxide, gold, aluminum oxide, so on. And then the frequency change is basically corresponding to the mass observed on the surface, which this event is the average event of the lot of molecule binding on the surface. However, by AFM, you can do exactly same thing, but it's a more precision fashion, which is you attach single peptide on the teeth. So those interaction of 130 piconewton, and then force-wise is very low force. So you can actually see how the interaction between those peptides with the substrate in the single level. And if you do the multiple time, you can have a very precision addition force, depending on how much you can actually do the force spectroscopy. In order to do that, you need to have a non-contact mode, very precision control by the like PAC system kind of material. So if you actually compare with the existing average signal with a single AFM signal, you can kind of see that the correlation is very nicely correlated. One is basically mass. So 30 Hertz meaning is 1 Hertz stands for the 17.7 nanogram per centimeter squared. However, you can clearly see that the single molecular force in a sense. So the first main result is basically we are investigating the absorption of the short random coil peptide with those inorganic substrate, and then we combining experimental approach allow us to compare the absorption kinetic and binding affinity. And using method uptake addition force, the gray value obtained from the bound SBPs on board, the result provide understanding of absorption behavior of the SBP on the solid support. So why this kind of technique or background technique is important? Because you can actually further modify now with a very functional molecule, because typically functional molecule containing very precise fashion. For example, in here, phosphoinositol is the biomolecule, which is one of the cell membrane components have known as a mediate key intercellular signaling pathway that controls cell survival, growth, disease, and so on. And if you actually see that this molecule is a different molecule, but it's very similar and complex. In this particular case, it's only different. Its phosphate group is changing its position. And depending on the 3, 4, 5 position, 4, 5 position, 4, 5 position, and 3, 4 position, and so on. So in order to target such a precision fashion, AFM is the most suitable technique in order to do that, because the specific binding of the protein and PI interaction is very, very important in order to capture the biologic process and fundamental idea and so on. And it is also existing in the HCV life cycle. Maybe you guys now are very interested because Hep C, we get them now this year, the Hep C got a Nobel Prize, as well as right now it's COVID-19 pandemic. So PIM molecule 4-5, typically important receptor also for the hepatitis C virus for the NSA5A domain. And then that's why people try to using this domain to develop the drug screening platform. So yeah, this is one of the example. So if you're using the AH peptide, how the addition for the specific and non-specific binding is accumulated with. And then this is done by the force spectroscopy interaction. So if you clearly see that if you have an AH peptide, you have a 183.8 plus minus 60.3 piconewton force. However, if it's non-specific, it's only 43. So it's a very distinct force you can actually capture very nicely with the AFM force microscope, force spectroscopy data. And then later on, if you control your density of the molecule within the system, you can clearly see that the force increasing as a function of density of this PAG molecule system. And then also we normalize how much we need to get the contact in order to how precisely we can actually capture and optimize the data in order to do the drug screening later on. So we can vary the either 0.1 second to the 2 second and 4 second and so on. So using that, depending on the contact time, 0.1, 1 second, 3 seconds, you clearly see how much is the force changes from the 54, 197 to 48. And we normalize as a 1 second contact time going to be the optimum contact time in order to see the very, very specific interaction in order to better understand the role of the electrostatic force in the PI binding, the effect of ionic strength also you can test it, and then as a function of a sodium chloride concentration. This is the physiologic sodium chloride. However, you can vary the sodium chloride in order to increasing, decreasing the non-specific interaction. So this is one of the techniques you can actually design the assay of the system. So here is what we do. So in order to screen the drug, basically, this is a picture here with the bilayer and the functional group, and then the team move with the mapping. So you can actually obtain the fraction receptor site by the PI binding small molecule. In this particular case, is neomycin, and decreasing the number of available reception site is available in this particular case. With that, we are able to actually using this force spectroscopy and using as a drug inhibitor on the PI. And then in this particular case, low concentration, no effect, similar addition force, and high concentration is the dose-dependent inhibition is very much possible. So you can actually position automatically. Therefore, automatically you can actually screening the drug, identify the molecule, screening the drug, and then you can also find that something can inhibit. So in this particular case, it's two of the example I'm mapping out. How do you actually using force spectroscopy, using non-contact mode, and then very precisely you can see the single molecular interaction. Control measurement parameter is the peptide coding density, which is the sample or drug or target molecular density, and contact time. And then also we study the influence of the ionic strength for the binding affinity, and then also using the drug candidate neomycin to able to demonstrate inhibit such an interaction. Therefore, you can actually identify the drug screening platform. Few of the advantages you can actually think about in this particular case. No more animal study, or prior to the animal study, you can precisely check with the biophysical technique like in AFM for spectroscopy, and that's going to be saved tons of time and precision because they can detect single molecular interaction. So whole conclusion is that binding affinity of those peptide with the inorganic material was studied by the force spectroscopy with the functionalized AFM tip. That was the background study, and then also we established a AFM platform to evaluate the interaction between peptide and then PI molecule. Also, we investigating the interaction between the NF5A, the BAP domain, and then PI receptor in this particular case. So with this whole study, you can guess that in this technique, maybe the future generation application for drug screening platform, because I know that POC system by FACS, they have not only the one T, they have a multiple T with the same force you can apply. The meaning is one single approach of the force spectroscopy, you're able to basically coating the different type of molecule, then you can have a precision fashion of how the drug you can actually screen, and then how to identify the drug screening platform and so on. So thank you very much for listening my talk, and then I'm very honored to be give you a talk. If you have any question, please let us know, and then I will answer the question. Thank you very much.